welcome to this class. In this class, I am going to explain you about the culture media, or you can say nutrient media. So let us start with the topic. So first of all, the question which arises here is, why is this culture media important to us? So this is important to us because it provides appropriate nutrients, pH, and growth conditions to the microorganisms. As you know, in microbiology, biotechnology, tissue culturing, we all need this culture media in order to support our specimen growth. Okay, but basically, since here I'm going to talk mainly about microbiology, so I'm going to explain everything in regards with microbiology only. Okay, so here let us start with the composition of the culture media. First of all, as I have explained to you earlier, that it is also called nutrient media. So basically what we are trying to grow here needs nutrients. Like if we are trying to grow some bacteria here, okay, so bacteria needs nutrients, and those nutrients are as follow. First of all, we add water to it, which provide hydrogen and oxygen. As you know that water's chemical formula is H2O, means two molecules of hydrogen and one molecule of oxygen. Okay, and then comes electrolyte. NaCl and KCl and some other electrolytes can be used, but mainly NaCl is used in culture media. The third which comes is peptone. Now peptone is partially digested protein. Okay, it is obtained from plant and animal source, and it is an intermediate form of protein that is digested partially. Okay, I have written same thing, sorry for that. And is used by microbe as a source of amino acid, or you can say protein, right? Now the fourth component is agar or geranium. It is obtained from seaweed, plant fungi. Okay, this is its source, and it is an inert solidifying agent that is used to make semi-solid and solid media. Then fifth and sixth are meat extract and yeast extract. Meat extract source is beef, and yeast extract source is Saccharomyces cerevisiae. Then comes the seventh point, which are additives. Okay, like blood and serum, which is added according to the requirement. Okay, now we will start with the types of culture media. The first type is of course solid media. Solid media is called solid because of its state. It is solid. Okay, we mainly grow colonies here. Okay, we do this streaking, spreading, and pore plating in the solid media. Then comes semi-solid media, which is mainly used to observe the motility of organism. Okay, and third is liquid media. The colonies grow in the liquid media in the form of turbidity, and this liquid media do not contain agar because agar solidifies the media. Semi-solid contains agar but in small quantity, let us say 0.5 to 0.8%, and solid media contain up to 2% of agar. Okay, that was on the basis of state, and now we are going to talk about the media on the basis of oxygen requirement, which is aerobic and anaerobic. The aerobic media are those media in which microbe grows in the presence of oxygen. Okay, and in anaerobic condition, oxygen is absent, and mainly in place of oxygen, CO2 is given. Okay, then comes the culture media on the basis of growth requirement, and this is the most important type of classification for culture media. Among them, the first is simple or basal media. Okay, this is the simplest media in which non-fastidious grow. Non-fastidious microorganism grow. See, fastidious. So, fastidious organisms are those organisms which need some special requirement. Okay, like some fastidious organism may require serum in it. Some may need blood, whole blood. Some may need only plasma. Some may need some factors like complementary factors and all. Okay. Then comes enriched media, enrichment media, selective media, differential media, transport media, and the seventh is complex media. Okay. So we are going to discuss about all these medias in detail right about now. And of course, we are going to start with the first media, which is basal media or simple media. Okay. So basically, I have written that it is used for performing biochemical tests like catalysis. Okay, but let us start with the introduction. Okay, so we have a fixed amount of, let us say, agars. We have a fixed amount of agar. After that, what happens is we just make our media out of it. Okay, so this simple media don't contain anything extra in it. It is just a simple media, just to grow microorganism. Okay, so this is a base preparation for any other media also. Okay, base prep. Like if we want to make complex media, this simple media is the base for that media, and we add into it, and then also you can say that other complex medias can be found. Okay, then comes the biochemical test like catalysis, like oxidases. These tests are also performed using this simple media. The second media which I am going to discuss here is, oh, I'm really sorry, is enriched media. Okay. Now this enriched media is a little special because it contains additives. Now what are those additives? Those additives can be blood, serum, or some other things like some salts, special salts regarding which are related to whatever microorganisms we are going to grow in it. Okay, so I'm going to discuss some media here. Okay, first I'm going to discuss about blood agar. So blood agar, it contains 5 to 10%, 5 to 10% blood plus nutrient media. Okay, so basically what happens is this blood agar is used to differentiate between three classes, which are alpha hemolytic, beta hemolytic, and gamma hemolytic bacteria. Okay, so these alpha hemolytic, these are partially they partially digest blood. Okay, so because they partially digest blood, what happens is, like, this is my petri plate and here is organism. So what happens is they will form a greenish color, something, not something, but a greenish color layer around themselves. Okay, I'm drawing it with red, but actually it can be green. uh, Let me just change the color. See, this is this green part. This is the characteristic of alpha hemolytic. Alpha hemolytic. Okay, and if we talk about beta hemolytic, then what happens is around there is a clear zone around the colonies. Okay, clear zone because they fully digest blood. Okay, since they fully digest blood, so the blood agar's blood will be digested around the street colonies. And about gamma hemolytic, they no effect because since they are non-hemolytic, that's why they don't have any effect on anything. In case of blood agar. Okay, let me now erase this, and I am going to explain about The second type of enriched media, it will take time, I'm really sorry. Okay, so second type of enriched media is chocolate agar. Chocolate agar. Okay, so in case of chocolate agar, what happens is that the blood agar is heated. So this is basically heated blood agar. Okay, so in heated blood agar, again 5 to 10% of blood is used in the nutrient, and then it is heated in 70 degrees Celsius, around 70 degrees Celsius. So as we heat it, so factor 4 and factor 5 are released out of this blood. Okay, and these are the components which are used to grow Haemophilus influenza virus. I'm saying virus, I'm really sorry. This is a bacteria, Haemophilus influenza bacteria. Okay, so this is how this fastidious thing grow. Okay, so after the chocolate agar, I'm going to talk about third one, which is biphasic media. Biphasic media. Okay, this is a special media which is grown in mainly in test tubes, in which what we do is one part of it is solid and other part of it is liquid. Okay, so since it contains agar and broth, so we can simultaneously watch the characters of the microorganisms. Okay, now these characters, how they grow in the solid, and how much turbidity they create in liquid media, it can be observed in biophasic media, which is a kind of enriched media. Okay, so after this comes fourth one, which is Loeffler's serum. Now in Loeffler's serum, it is a special media which is used to grow Corynebacterium diphtherium. Corynebacterium diphtheria. Okay, so it contains serum as enrichment. Okay, so this Loeffler's media is enriched with blood serum, so that Corynebacterium diphtheria can grow in it. Okay, and the next media which comes into play, I'm really sorry, which comes into play is enrichment media. This is a kind of enriched media, but it is mainly found in the form of liquid. Okay, liquid broth. So this, since this is in the form of broth, so you can say it is a little different from enriched media, which is in the form of solid or semi-solid, and can be biphasic also. Okay, so here this enrichment media is used to isolate pathogens from stool and sputum sample. Okay, stool and sputum sample. Stool and sputum samples are mainly those samples uh, from which bacteria or microorganism is isolated using enrichment media. About enrichment media. Okay, so basically stool and sputum samples are the samples which are used to isolate bacteria from stool and sputum samples using enrichment media. Okay, and some examples of enrichment media are like tetrathione broth. Tetrathione broth. Now this particular enrichment media is used to grow Salmonella typhi. Okay, then other is alkaline peptone water. Okay, this alkaline peptone water is used to grow Vibrio cholerae, right? So that was our enrichment media, and now I'm going to move to the selective media. As the name suggests, this selective media is the media which selectively in which we can selectively grow an organism. Selectively grow. I'm writing microorganism here. Okay, MO means microorganism. So there are some examples of the media like Lewinson Jensen LJ media. Okay, um, I don't know spelling of this Lewinson. Okay, Jensen is Jensen. So this particular media is used to isolate Mycobacterium tuberculosis. And second is thiosulfate citrate bile salt agar. Thiosulfate citrate bile salt agar. Now this media is used in isolation of Vibrio species. Okay. Then comes Wilson Blair bismuth sulfide media or Wilson and Blair media. Wilson Blair bismuth sulfide media. Now this particular media will isolate Salmonella typhi species selectively. Okay. Then comes differential media. Ink color. Okay. So differential media is not that different from selective media. It's just that it differentiate two major groups. Differentiate two major groups. Now, for example, McConkey agar. Now, this McConkey agar, if we add methylene blue, we can differentiate fermenting bacteria and non-fermenting bacteria. Okay. Similarly, this can also differentiate between gram positive and gram negative bacteria also. Then comes transport media. Mm, okay. So basically, this media is a kind of preservative media. So basically, when we have to transport our microorganism, let us say from one lab to some second lab. At that time, this preservative media or transport media comes into play. Okay. So some example of this transport media are like Venkatrama Ramakrishnan media. Okay, this particular media was invented by an Indian scientist, as you can hear from the name, and this is a transport media. Okay, so that's how some transport media like Carrie Blair media is also there. Carrie Blair media, then Amy's and Stewart media. These medias are also there which act as transport media. Okay, then oh, I'm really sorry. Then comes our complex media. Now this is a non-routine media. You can say this complex media is basal media plus some enrichments. Okay, so this complex media is different from our routine medias. Like whichever medias you have read before this, they all comes in routine category. Okay, routine category means you can get a box. Like let us say I am getting this particular box from industry, and that industry they earlier they have added every component into this box. So all we have to do is read the instruction out of that box and make our media. But this complex media is different because this is a media which we make in lab. Okay, we make this complex media in lab in order to grow some microorganism which cannot be grown in our routine medias. Okay, So that's why this complex media is used. Basically these are only the introduction for our medias, and you can obviously write these in exams also using some reference books and my lectures. That's it for this class. Thank you very much and see you soon.